Good afternoon, everyone. So welcome again to the ILC Symposium, and now in the last session of this symposium. Thank you for staying with us. So today, during this session, we will learn much more still about the biology, actually, of lobular tumors. And we will start the session with Professor Val Brunton. So Professor Val Brunton is actually a principal investigator and a professor of cancer therapeutics at Cancer Research UK in Edinburgh. She has a strong interest in the signaling and the regulation of cancer growth and metastases. She's also a member of ELBCC. And as I introduced you yesterday during my keynote lecture, today she will tell us more about the specific features of the tumor microenvironment of lobular tumors and how these can be different from the ductal tumors. So I'm very much looking forward to listen to your story. And thank you for accepting to present. Val, it's up to you.
Okay, thank you, Christine, for the introduction and also for the invitation to talk to you all today. So we've heard throughout this meeting that the ILC is a unique clinical and molecular entity, and we now know a lot about the molecular makeup of lobular tumours. In contrast, we know relatively little about the tumour microenvironment in these lobular tumours. And more importantly, we don't have any real idea of how the tumour microenvironment might be influencing some of these unique features of the lobular tumours, such as the resistance to chemotherapy or the dormancy that is seen in lobular tumours, or the unusual metastatic spread. So Christine and colleagues, as you heard yesterday, have now defined many of the changes that occur within the immune microenvironment in lobular tumours. But we don't really know very much about the other stromal components, such as the cancer-associated fibroblasts and also the extracellular matrix composition. What we do know is that lobular tumours, due to their infiltrative growth pattern, have a very extensive stromal component. And now the collagen architecture and deposition in lobular tumours has been recently characterised and quantified and compared with other tumour types. And also we heard in the earlier sessions today, very nice work from George Thomas and workers who have identified a tumour intrinsic regulation of the lobular extracellular matrix composition within the tumour microenvironment, and have identified a potential target in the tumour microenvironment of the lobular tumours in the LOX enzymes. But we were interested in what the role of the cancer-associated fibroblasts are in the lobular tumours. So cancer-associated fibroblasts are actually the predominant source of extracellular matrix proteins and collagens within the tumour microenvironment. They also have a range of other pro-tumorigenic roles within tumours. They secrete growth factors and cytokines, which have pro-survival and migratory activities for tumour cells. They're also important for recruitment and activation of immune cells. But really, we know very little about cancer-associated fibroblasts in lobular tumours. So we've started to work on this, and this is just work from work that we've been doing in the KEP model. So the KEP model is a genetically engineered mouse model of lobular breast cancer, which is driven by a loss of E-cadherin and a loss of P53 in the mammary epithelium. And what we did in this experiment shown here is that we isolated both epithelial cells and fibroblasts from these KEP tumours, and we reintroduced them into the fat pad of animals. And we can see that when we reintroduced the epithelial cells in the presence of the cancer-associated fibroblasts, that we got a dramatic increase in tumorigenicity in this situation. So we were interested to understand more about how the cancer-associated fibroblasts may be different in lobular tumours compared to ductal tumours. So we started a programme of generating primary cancer-associated fibroblast cell lines from tumours, both of ILC tumours and also ER-positive ductal tumours. And we've been generating RNA-seq data from these primary cancer-associated fibroblasts. And In the analysis that we've done so far, you can see on the heat map here, we've identified this analysis identified 156 differentially expressed genes between the ductal derived and the lobular derived cancer-associated fibroblasts. When we carried out, when we looked at this further and looked at the genes that were differentially regulated, we found enrichment analysis showed us that genes that were involved in MYC targets and mTORC signaling were enriched in the lobular derived cancer-associated fibroblasts, whereas genes involved in TNF and NF-kappa B signaling and hypoxia were enriched in the ductal derived cancer-associated fibroblasts. So as one of the main functions of fibroblasts is to induce paracrine signaling through secretion of growth factors and cytokines, we carried out an experiment where we took conditioned medium from some of our cancer-associated fibroblast cell lines, and we treated the SUM44PE lobular breast cancer cell line with the cancer-associated fibroblast conditioned medium and looked at signaling changes in the SUMs using an RPPA analysis. And we can see here in the heat map that we saw changes in a number of different signaling pathways. And in the graph, we can see that the most significant changes that we saw in the SUM44s was activation of pathways linked to ERK signaling and also STAT signaling, STAT1 and STAT3 phosphorylation. Interestingly, when we then went to look at the RPPA data that's in the TCGA data set, and we compared ER-positive lobular tumours with ductal tumours, we found that these same signals were increased in the lobular tumours compared to the ductal tumours. We were particularly interested in this increase in activation of STAT3 that we saw in the lobular tumours. STAT3 is an important transcription factor, and it's known to have a number of pro-tumorigenic activities in different tumor types, and especially being reported in breast cancers. So first of all, we decided we were going to look at this in a little bit more detail, and I looked at known regulators of STAT3 activation in our cancer fibroblast data set, and we identified IL6 as being potentially important for inducing this activation of this pathway. IL6 is a very well known and recognized activator of STAT3. So what we did was we confirmed using an ELISA that actually the IL6 protein was secreted by the cancer-associated fibroblasts. You can see here secretion in the different fibroblast cell lines that we tested, but no secretion from the ILC epithelial cell lines. And then when we looked at this experiment here, where we've actually used a blocking antibody to anti-IL6, we could see that the increase in STAT phosphorylation that we saw in the SUM44s upon addition of conditioned medium, this just shows conditions with two different CAF cell lines, that we could effectively inhibit this activation of STAT3 when we had the blocking antibody there. Interestingly, we also saw the activation of ERK signaling that we saw following stimulation with a conditioned medium from the calves. We could also see a reduction in activation of this pathway with the anti-IL6 antibody. We also looked at IL6 expression in bulk tumor sets. This is from the TCGA data set, and we saw that IL6 was increased in lobular tumors compared to ER-positive ductal tumors. And then we went back to the TCGA RPPA data set, which had where we'd seen the changes in the phosphorylated STAT protein, and in the samples where we then compared IL6 expression with activation of STAT3, and we could see a very nice correlation between the presence of IL6 and the phosphorylation or activation of this STAT3 pathway. So we're now following this up and with some mechanistic studies looking at how this IL6 STAT3 signaling access might be important in some of the functions of the lobular tumors. So we'd also generated a second data set where we'd use laser capture microdissection to separate out the stromal and epithelial components from a number of ILC tumours. And so here, when we dissected out the stroma, what we were taking out was predominantly the extracellular matrix and the cancer-associated fibroblasts, and we were trying to avoid dissection of the immune cells. And you can see here in the heat map, we've got very nice separation of the tumour epithelium from the tumour stroma. And as you would expect from the way that we carried this out, we've got an enrichment of genes associated with matrix organization and collagen degradation, integrin signaling in the stromal compartment, whereas in the epithelial compartment, we've got an enrichment of Rho GTPase effectors, also with genes involved in cell cycle and DNA methylation. So a nice separation of the tumour and epithelial stroma in this data set. And we then went on to carry out a series of analyses which we were wanting to identify genes that were specifically enriched in the stroma of the lobular tumours compared to ductal tumours or normal breast. So we did this in collaboration with Morag Park and Nick Bertus at McGill University in Montreal, because they had previously generated data sets from ductal tumours and also from normal breast. And what this allowed us to do was compare our data set with the ones that they'd already generated, and we identified 261 genes that were upregulated only in the tumour stroma of the ILC tumours. We carried out another series of analyses, and this included also filtering for genes that were only expressed in our CAF data sets, and also we looked at which of the genes were increased in bulk tumour data sets, the TCGA and metabric cohorts, for genes that were overexpressed in ILC tumours compared to estrogen receptor positive ductal tumours. And this resulted in a list of 10 genes, which you can see here were enriched in the stroma of the ILC tumours, but not the epithelial cells shown here, and were not enriched in the stroma in the IDC or normal tumours. And so this is the list here. It contains a number of matrix protein collagens, tenacin C, also some matrix regulation proteins, MMP2s, TIMP2s, and also a couple of signaling proteins. So what we wanted to address was whether any of these genes that we've identified in this data set, and also in the primary CAF data set, whether any of these genes were associated with survival. So the majority of them were not associated with survival when we looked at this, and so we were comparing ILC and IDC tumours in a number of breast cancer cohorts. And this shows you the data that we got for TIMP2, where we did see that high levels of TIMP2 were associated with a better prognosis in ILC tumours, but we didn't see the same association in ER-positive ductal tumours. So TIMP2 is a tissue inhibitor of metalloproteinases. It specifically inhibits the activity of MM2, which is a key collagenase within tumours. So MMP2 will break down collagens within the tumour and promote tumour migration and invasion. And when we did a network analysis of the genes from both of the data sets that we looked at, we can see here that we various interconnected genes, and we have TIMP2 here associated with MM2, which we also picked up in the laser captured dissection data set, and also another matrix metalloproteinase, MMP16, which we actually picked up as one of the genes in our enriched in the CAF gene data set. And so what we're doing now is we're actually just looking at whether we can validate this effects of TIMP2 at the protein level in lobular tumours. So actually the most highly enriched gene that we found in the laser capture data set was this gene here, PAP-A. So And this is the data shown here in a little bit more detail. We see an increase in the tumor-stroma, but in the lobular tumors, but not in the ductal or normal. We're also able to show this in further ILC tumors using RNA scope. This just shows you here the change in the individual tumors and the overall difference between the tumor and stromal expression. So PAPA encodes this protein called pregnancy-associated plasma protein A. And PAPA is involved in insulin growth factor signaling pathways. So it's a secreted metalloproteinase, and it cleaves insulin growth factor binding proteins. And by doing this, it releases active, bioactive insulin growth factor, which then can act on its receptor, the IGF-1 receptor, and promote IGF-1 signaling in the target cells. The activity of PAPA itself is actually strictly regulated within cells and tissues. It binds to staniocalcins 1 and 2, which inhibit its activity. And as the name suggests, PAPA was initially identified as a protein that was increased in expression during pregnancy, but now it's been shown to be widely expressed in a number of different cell types and also in a number of different epithelial tumors, including breast cancers. And here it's been reported to be expressed predominantly in the epithelial tumor cells themselves and not the stromal cells. And in fact, there are efforts ongoing to develop anti-PAPA blocking antibodies based on promising preclinical data showing that it's able to block tumor growth in a number of different models. So we were interested in understanding whether in the lobular context, whether it could be acting in a paracrine fashion. So first of all, we looked at PAPA expression in a panel, a series of breast cancer cell lines. As you've heard earlier, there's very few lobular cancer cell lines, but when we did look at the ones in this panel, the MDA MB134s and the SUM44s, we saw that PAPA was not expressed in these two cell lines, but actually more generally, when we looked across the panel, there was low or absent expression of PAPA in the majority of the luminal breast cancer cell lines. This just shows some validation using PCR. We can see no expression of PAPA in the luminal cell lines, but we did see expression in all of the fibroblast cell lines that we've looked at. We also were able to validate that the PAPA protein was secreted from the fibroblast. This is an ELISA assay that we carried out. So we can see here PAPA secreted into the conditioned region from the cancer-associated fibroblast, but not from the epithelial cells. As I mentioned, the activity of PAPA is tightly regulated by binding to these staniocalcins 1 and 2. So this assay allowed us to ask whether the secreted PAPA was actually active. So we've treated recombinant IGF binding protein 4 with conditioned medium from the cancer-associated fibroblast, and the lower band here is the cleavage fragment of IGF BP4. We can see here in the presence of conditioned medium that you have a cleavage of the intact IGF BP4, and if you pre-treat with the PAPA inhibitory antibody, that you can reduce this, and this is just a second repeat here. So I don't have time to show all the data here, but what we think is happening in the context of these lobular models is that both the PAPA and the IGF-1 are being secreted by the cancer-associated fibroblasts, and this leads to a release of bioactive IGF-1, which then can act on the IGF-1 receptor in tumor cells and induce downstream signaling in the tumor cells. So this was of particular interest because previous work had also shown that E-cadherin loss is able to regulate IGF-1 signaling and increase and leads to an increase in signaling through the IGF-1 receptor. And IGF-1 itself is also increased in ILC tumors. And also, it's been shown that loss of E-cadherin enhances the sensitivity of breast cancer cells to anti-IGF-1 receptor inhibitors. So it could possibly be that the PAP-A signaling axis is another mechanism whereby these tumors could increase IGF-1 signaling. When we actually looked in bulk tumor data sets, we found that actually PAP-A was higher in cadherin negative tumors than in cadherin positive tumors. But actually, this is probably driven by an increase in expression in the Claudin low subtype, as this is a subtype that we found to have the highest levels of PAP-A expression. However, it wasn't quite as the story got slightly more complicated when we looked at survival and the role of PAP-A in survival of breast cancer cohorts, because actually what we found in lobular tumors was that either high or intermediate levels of PAP-A were associated with a better survival in the lobular tumors. And interestingly, we didn't see the same effect when we looked at ER-positive ductal tumors. So although we have a nice correlation between the IGF-1 gene expression and PAP-A gene expression in the lobulars, which we don't see in the ductals or the normal, when we went back to the TCA data set and looked at expression of the phosphorylated IGF-1 receptor, which we're using as a surrogate of pathway activation, we see in the matched samples when we looked at PAP-A expression that we see no correlation between the expression of PAP-A and pathway activation. So I think this really reflects that in tumors there's a very complex regulation of this PAP-A IGF signaling axis, which I think you don't see in normal tissues, because there's very good evidence in normal tissues and and in mouse models that actually the PAP-A signaling axis does play a really important role in activating IGF-1 signaling. So a lot more to be done to understand really the role of PAP-A in not only in lobular tumors, but in the wider context of breast cancers. So I'd just like to finish there and thank the people that have been involved in this project. Laura and Esme are two PhD students who have carried out the majority of the work that I've shown here today. Also like to thank our clinical colleagues and patients in Edinburgh who have made it possible for us to access the clinical material that we've used in these studies. Also, Morag Park and Nick Bertos at McGill for collaborating on the laser capture microdissection study. At the Institute of Cancer Research, Rachel and Syed have been carried out the survival analysis studies that I showed, and then Klaus and Per Neil have been involved in the PAP-A story. Finally, I'd just like to acknowledge the contribution of Andy Sims to this project. Andy sadly died of melanoma last month, and we started this project many years ago together, and he was a great collaborator and also an inspirational supervisor for both Laura and Esme and his other students, and really without him this project wouldn't have happened. So I'd just like to finish there, and I'm happy to take any questions.
Okay, thank you very much, Val, for this beautiful, beautiful presentation. I'll have a look in the questions, but I think my question chat is blocked. So I'll start with some questions myself, then if you allow me. So actually, if we go back to the beginning of your presentation, when you were actually isolating cancer-associated fibroblasts from ductal and lobular patients, did you actually focus only on tumors that were estrogen receptor positive and HER2 negative, or didn't you consider these variables?
Yes, they were all ER positive, and we tried to match them. They're all grade 2 tumors that I think they're all grade 2, yeah. And so yeah, we did try to match them. I think one of the lobular cell lines was HER2 positive. But yes, we were conscious that we were trying to sort of reduce the variability when we were comparing the ductals and the lobulars by trying to match them as much as possible when we got the clinical material. Yeah.
Okay, thank you. I now have, it seems. I also now have a question actually from the audience. So the question is, how can IGF inhibitors affect clinical practice in ILC? Are these drugs like the approved metformin?
So yeah, I mean, the IGF-1 receptor inhibitors have been used, have been trialed a lot in breast cancer. There's been a number of different companies have IGF-1 receptor inhibitors, and they've been through extensive clinical trials, but really have been quite disappointing in terms of their activity. And so there's a lot of questions about their specificity, because they also they also inhibit the insulin receptor as well. So there are some toxicities associated with them. So it's not clear exactly that we know exactly what the biomarkers are that would predict which patients would respond to an IGF-1 receptor inhibitor. And I think that's a problem at the moment. The idea with the PAP-A blocking antibodies that people are trying to promote is that this might be an alternative use of to IGF-1 receptor inhibitors, but again, we still wouldn't know exactly which patients would benefit from this. And from our work with the PAP-A looking at the survival, it's not exactly clear how that pathway feeds into the IGF signaling in the lobular tumors.
Okay, thank you. Another question from the audience is the following. So in the CAF conditioned medium experiments, do you see similar effects and their levels in CAF derived from classical versus pleomorphic ILC? So did you focus actually on the different histological subtypes within lobular tumors to see whether there would be different effects?
Yeah, we haven't got that level of differentiation in the CAF lines. That we haven't got enough CAF lines to make any comment on that at the moment. Yeah. I mean, we do obviously know which subtype they came from, but yeah, we don't have the numbers to make any comment on that.
Okay, thank you. Another question here from Patrick. So most IGF-1 is produced by the liver, which you will miss with mRNA analysis. Have you looked at IGF-1 serum levels in these patients actually with ILC versus IDC?
Yes, we haven't done any of that, but there are lots of other studies which have looked at serum levels of IGF-1 and also the IGF binding proteins. I'm not sure if that has been done in the context of lobular tumors though, but definitely in wider context of breast cancer. Yeah, so there is evidence out there for increased levels of IGF-1 and the binding proteins, and people have looked at that in terms of prognosis.
Okay, thank you. I also had 2 other questions, and the audience still has 2 minutes to also ask questions if they want. So I was wondering whether you were also not only comparing the quality and the transcriptomic profiles of these CAFs, but also maybe whether the amount was different actually between lobular tumors and ductal tumors.
Yeah, so we haven't. Yeah, that's been a sort of. We've been thinking about the best way to do that for a while. Yes, we haven't actually done that. I mean, there's a little bit of evidence out there that there are increased numbers of CAFs in there, but I think because of the heterogeneity that you see in the different CAF populations, I think it would be, it'd be better to think of that in the context of also trying to sort of understand whether there was different subpopulations as well. So I think this is something that I think they're looking into in Paris. I think they're starting to do those sort of studies now.
Yes, and then the last question. So we know that cancer-associated fibroblasts are, as you said, a very heterogeneous population. Also that they can have different origins. Do you think that there would be a different origin for this cancer-associated fibroblast in lobular versus ductal tumor?
Oh, yeah, interesting question. Yeah, I have to say, I don't know. I wouldn't. I don't know whether they would or not. Yeah, I haven't really thought about that, to be honest.
Okay, but thanks again for this beautiful presentation, and we will now move actually to the next speaker of this cancer biology session. So we'll now move to Professor Adrian Lee. So Adrian Lee is actually Professor of Pharmacology, Chemical Biology, and Human Genetics at the University of Pittsburgh. He's also the Director of the Institute for Precision Medicine, and he's actually the partner in crime of Steffi Oosterreich, because they are leading the lab together, the Lee Oosterreich lab. And their lab actually studies the molecular basis of breast cancer development and resistance to therapy, with the goal to improve precision medicine and outcomes for breast cancer patients. And they have a very special focus on the understanding of invasive lobular breast cancer. And today we really look forward to listen about their study about the single cell transcriptomic heterogeneity in invasive ductal versus lobular breast cancer cells. So Adrian, the screen is yours.
Thank you, Christine. And you can see my slides okay?
It's perfect.
Okay, great. Thanks, Christine, for the introduction, and thanks to you for the organizers for the opportunity to present today. These are my disclosures. The biggest one, as you know, as Christina just said, I'm married to Steffi, and that may have had an impact upon my choosing to be a presenter here today. So I won't give an introduction to ILC, because we've had a series of, over the last 3 days, some fantastic talks about the background to the unique clinical and molecular features of ILC, but I thought I would tell you about what I'll focus on today, which is single cell RNA sequencing, with just really one background slide showing you a very nice Nature Protocols review on this, showing the evolution of the methods to do sequencing of individual cells so that we can understand the heterogeneity of cells within a tumor, but then the ability to do large numbers of cells. So this is a log scale, numbers of cells sequenced, and this is time. And you see just in the last 5 years, there's this exponential growth in the ability to sequence individual cells, where now you can sequence up to millions of cells. I show this at the level of RNA sequencing, which is what I'll speak about today, but basically you can do this at the level of DNA sequencing, methylation, ChIP-seq. Really, in the end, probably everything we'll be able to do at the single cell resolution. This, I think it was Dr. Jesselson who said yesterday, is really going to be transformative for our understanding of cancer, because it's going to be like going before the microscope, where you had all this kind of black bile and stuff, to after the microscope, where you could see microbes. Now we're going to see, with the resolution at single cell, the real heterogeneity, and try to unravel the complexity of cancer. So I broke this down into 4 parts. I will speak about single cell RNA sequencing of cell lines, which was published about 6 months ago. I'll just show you one slide on that, because this is already published, and the rest will be unpublished and work that's mostly in progress on sequencing of patient-derived organoids, sequencing of mixed IDC, ILC, and then single cell sequencing of bone metastasis. So as I said, this was just recently published. Our first foray into this was just simply, you've heard from many of the talks, these ILC cell lines that are available here. We took IDC cell lines as a reference, ILC cell lines, mimicry, where we genome deleted CDH1, and then 2 other non-transformed cell lines, mixed them all together, did single cell RNA sequencing, and then deconvoluted them and said, what's the difference between them? So when you do that, each dot on here represents an individual cell with its whole transcriptome. And these cells, which are colored here, cluster very differently. So the cell lines all look very different. So that's great. So that means we can split them apart, and we can then start to interrogate the underlying principles of those cell lines. The non-transformed cell lines are found here at the top. The ILC cell lines cluster here in the middle, and the IDC cell lines cluster down here at the bottom. What was interesting was this here. So basically, as I said, we genome deleted E-cadherin or CDH1 from T47D cells, an IDC cell line. You can see this is E-cadherin. This is where it's been genome deleted. They start to look like ILC. They have these lack of cell attachments. They grow grape-like when you grow them in 3D, like Patrick had said earlier. And when you do single-cell sequencing, you can see that they now cluster very differently. So this is the T47D wild type, and this is the T47D without E-cadherin. So just simple deletion of that one gene really reprograms the transcriptome for those cells to look quite different, and in part explains why it's a major driver of this disease. This is the only thing I'm going to tell you about this paper. You're welcome to go and read it. In that paper, we drilled very deep into the heterogeneity within the cell lines. And in the ILC, we also identify a putative dormant estrogen independent subclone in MCF7s. And then we identify an IRF1 regulatory network that seems to be uniquely increased in ILC cell lines and ILC tumors. But what's been mentioned multiple times, actually Val said this just now in her talk, and then George Flomos said this earlier today, is that we just have very few models. Everyone uses the same 3 to 4 models. And I should say, before I go on to the next slide, this data is all publicly available. So if you go to Geo, you can download it, and you can do everything yourself, and you can look for your genes of interest. So there are very few ILC cell lines, and this shows the extent of the problem. This is PubMed entries for IDC cell lines versus ILC cell lines. And you can see this is again on a log scale. So MCF7s is up here with about 30,000 publications. And the ILC cell lines, as you see, are really understudied and then underpublished. 3 of them don't have any publications because they're not out yet. One of this is a new cell line we've generated, WCLC25, which we haven't yet published on or reported. But even the ones that are reported, there's just very few publications on them. And this is a problem for the field that is well recognized. As an example, the Cancer Cell Line Encyclopedia, which is one of the kind of data dictionaries of cancer cell lines, only had 2 ILC cell lines in it. So basically, we don't know much about these ILC cell lines. Interestingly, the Rather study had reported that some of these IDC cell lines actually have E-cadherin mutations. And some of these E-cadherin mutations that are in these putative IDC cell lines actually look like ILC mutations. And it's possible that some of these cell lines, many of them were made like 30 years ago, and some of them are just labeled as carcinoma, that there was no annotation at the time. Someone wasn't thinking about ILC versus IDC. They just said it's a carcinoma, breast carcinoma. But in that paper, they didn't really study these ILC-like cell lines any further. So what we've done is to gather all of these ILC cell lines, all of these cell lines, and do a very comprehensive characterization of them and credentialing of them that I think will help researchers choose better models for study. So this is something called we named the ILC Cell Line Encyclopedia, ICLE. This is running off of the Cancer Cell Line Encyclopedia. As I said, we took all the ILC models. Some collaborators gave us them. Others are publicly available. We took all the ILC likes. This tells you the ER and HER2 status. We took unmodified cell lines, authenticated them, got them from vendors, and now have done and completed pretty much every omics analysis you could do: methylation, optical mapping for structural variants, SNP arrays, whole exome sequencing, mRNA and microRNA sequencing, reverse phase proteomics, and then immunofluorescence. This is done as part of the NCI Oncology Models Consortium and in collaboration with LBCA. We are about halfway through the phenotypic characterization, looking at migration, estrogen response, and growth, because we think this will be important for integration to the omics and then for people to choose their models of interest. And we collaborate through this consortium with the Broad Institute to do loss of function and drug screening through RNAi CRISPR and a drug sensitivity screen. So at the end of this, investigators will have a choice of a much larger series of models of which they can pick their omics features, their phenotypic features, their drug response features, as you can do in ductal cancer. You can clearly do that in ductal cancer at the moment. And I'll just show you, as I said, the omics is all finished and we're working through this. It's quite a lot of data. I'll just show you one feature which I think is very encouraging, and that's that these cell lines recapitulate the CDH1 mutation landscape of ILC in patients. So this is patient tumors from the Cancer Genome Atlas, looking at mutations. All the mutations, nearly all mutations are enriched of CDH1 in ILC, as we know. The mRNA expression of E-cadherin is low in ILC, copy number is deleted, and the protein in blue is very low compared to IDC. This is published and well known. These are these cell lines that we've now been working on. So this is IDC cell lines, ILC-like from the RAVA consortium, and then the ILC that I showed you, the 8. You can see again, the CDH1 mutations are enriched in the ILC and ILC-like. The ECAD expression is low. They have copy number loss. And again, the protein is low. And these ILC-likes actually look very similar to the ILCs. And they have mutations which pretty much guarantee that these are likely ILC that were never annotated when those cell lines were made many years ago. When you look at the CDH1 mutations, they're very, like I said, they're very indicative of ILC. So these ILC-likes have these truncating frameshift mutations in CDH1, which are never found, nearly never found in invasive ductal cancer. Invasive ductal cancer cell lines only have these missense mutations that are likely passenger mutations that don't affect E-cadherin. And then you can see this here. So this is looking at these truncating mutations. In ILC, there's a large number of truncating mutations. You don't find those in IDC, in human tumors. Similarly, in these cell lines, the truncating frameshift mutations are only found in these cell lines. They're never found in the invasive ductal cancer cell line. So I think this is going to be, once we finish this, an incredible resource to do better selection for models. I should as well say that these biallelic mutations, which is the majority of ILC, have both LOH and point mutations. They're nearly always found in ILC, and that's how we know that these are probably ILCs. So we all know the problem with growing cells on plastic. These cell lines have a huge number of problems, and so we've been focusing on making ILC patient-derived organoids and sequencing of those, and I'll show you this now. So organoids have become very popular since Clever's really, kind of stimulated the field to do this, and we follow a protocol from Clever's nearly identically. We get tumor tissue from the clinic, mince it and digest it, grow it in this. It's a relatively simple protocol once you've learned how to do it. And you can now grow these organoids. We have about an 80% success rate to grow organoids. And you can see here, this is an invasive ductal cancer organoid. You see it's quite tight and compact. It expresses E-cadherin. These are these ILC organoids. So here's a couple of them here. And you see they have this unique grape-like structure, which Clever's actually, their group actually reported in the original publication that the ILC looks very different to the IDC, and of course, doesn't contain E-cadherin. So we have attempted about over 100 or 120 organoid developments from human tissue, from rat models, from mouse models, and in the humans, we've done it from normal breast, from premalignant disease, from invasive cancer. And we have 85 successful human patient-derived organoids. Over 40 of these are from invasive ductal cancer, and over 20 of these we've enriched to get the number up for the invasive lobular cancers. Similar to what I said about the cell lines, these truncated frameshift mutations in E-cadherin are exclusive to the ILC patient-derived organoids. You don't find these in the invasive ductal cancer organoids. So this indicates their ILC nature. So we've been using single-cell sequencing because one of the features of the organoids is they have a heterogeneity that you just don't have in cell lines. They lose heterogeneity when cell lines go onto plastic, whereas the organoids retain that. And to analyze it, you have to look at single-cell sequencing. So this is a single-cell sequencing of a tumor. This is an ILC metastasis. You can see all different cell types here. When we put this into organoid culture, this is looking at the tumor RNA from the metastasis versus the organoid. You can see it's nearly perfectly correlated. This faithfully represents the RNA that was present in the original tumor. And you also get the heterogeneity of clones in there. So this is tumor versus organoid, 3 different patients here. And you can see different clones, epithelial clones found in the tumor, are nearly always represented in the resultant organoid. You do not see this in cell lines. And as proof that this doesn't happen in cell lines, this is comparing the numbers of clones or heterogeneity of an organoid versus heterogeneity of a cell line. And you see cell lines have much lower heterogeneity than the organoids do. So they will be superior in that regard. The other thing is we need models, estrogen receptor positive models. Nearly all of ILC is estrogen receptor positive, and some of these have very good estrogen response. So this is 2 organoids here. This one is estrogen receptor negative. This one is estrogen receptor positive. We can see estrogen receptor by staining. We do single cell sequencing, and the estrogen receptor negative ones have very little cells positive for estrogen receptor at the RNA level. These 3 you can see have nice, robust expression of estrogen receptor in many of the cells, and they have a robust estrogen response. So this is looking at estrogen induction of GREB1 and estrogen induction of TIF1. You see these 3 here, which are ER positive, have nice induction. And actually, when you do this comparable to MCF7s, they have a greater induction than we see in the MCF7 cell line, which is the positive control. So I'll give you a little bit of a hint about what we're doing on the single cell sequencing of mixed IDC. This is a mixed IDC ILC is a bag of a spectrum of diseases that aren't IDC and aren't ILC, and they get put in together. And they're incredibly complex. And I think we thought this would be the key area where single cell sequencing will let us understand the different cell types that are found in here. And we have now done 3 of these, and I'll just show you one of them. And this shows you the complexity of the disease. So this is a classic mixed IDC ILC. You have a ductal component here. You have a lobular component that's E-cadherin negative. This is E-cadherin positive. Over here, there's a ductal component now that's E-cadherin negative, so that's pretty confusing, and the lobular component. Here you have just a lobular component, and here you have a ductal component, yet the E-cadherin is kind of aberrant. It's not present like it is here, and it's not absent like it is here. It just seems to have this weird expression. So you have this incredible complexity of cellular and histomorphologies that single cell sequencing, we thought, might help unravel that. And not surprisingly, the data is super complex, so this is underway. So I'm just showing you pilot data. But in this patient, we again do single cell sequencing. We identify T cells, endothelial cells, fibroblasts, and we can identify cells, 3 populations that probably are the ductal component, which is CDH1 positive, and 3 populations that are CDH1 low and probably are the lobular component. These can be broken down by various other biomarkers such as progesterone receptor and various keratins. And you can visualize this here. This is the keratin 19 positive population, which is the epithelial population here. These are non-epithelial. And in that epithelial population, you see ones that are CDH1 positive, and you see cells which are CDH1 negative. So presumably there's some differentiation there between them. And I won't go deep into this, but basically you can then look at copy number changes, and basically there's a lot of heterogeneity in the copy number populations that we think at the DNA level will then help us unravel the lineage of these different populations of cells that are arising in the keratin population. So I'll end with how do you put all of this together? Can we take all of this, put this together, and use this for how we believe we should be enabling the treatment of patients with invasive lobular cancer? And I'll show you just one use case of a patient with invasive lobular bone metastasis. So we are focused on trying to use all the tools we can to understand why primary disease metastasizes, applying whole exome sequencing, RNA sequencing, single cell sequencing, and then organoid development. And so this is an example of a 74-year-old woman who had estrogen receptor positive, progesterone receptor negative, HER2 negative ILC, stage 3C. She received a lymph node dissection and a segmental mastectomy, had chemotherapy plus aromatase inhibitor, very standard of care. Unfortunately, 44 months after this diagnosis, she had a recurrence. She had metastasis to the left bone. This is the hip region on the left and the tibia on the right. She had resection of those, received extra therapies, and then unfortunately has now passed away. And we took this opportunity to try and understand what had happened during this disease progression and whether single cell sequencing and organoids could help us understand that disease progression. So when we look at this case, we can see this classic evolution, which is the complicated nature of this disease. So this is the primary disease here, the primary breast cancer, And then this is the bone metastasis on the left and the bone metastasis on the right. And you can see when you look at gene expression, there's increase in PI3 kinase, AKT, and mTOR signaling when they metastasize. There's increased cell proliferation and antigen response. There's unique signatures. The bone metastasis here on the left, bone metastasis 1, has this increase in hypoxia and angiogenesis in EMT. You don't see that on the right. And then there are some signals that are lost. So you lose estrogen receptor signaling because this actually is a tumor that went from ER positive to ER negative, and probably is one of the reasons why it recurred while on endocrine therapy. At the DNA level, there was also changes in DNA mutations. So a PI3 kinase mutation was found, a driver mutation in the primary and the 2 bone metastasis. There were 37 common mutations. However, not found in the primary, but in both of those bone metastases was a BRCA1 somatic mutation. So this is gained in the metastasis. And then there were a bunch of mutations that are unique to each side of the bone metastasis, reflecting the complexity of the disease that woman now has at the point of metastatic disease. So we have organoids grown from all of these, and now you can treat them. So the organoids have these 2 mutations, the PI3 kinase and the PARP inhibitor, and you can see they are very sensitive to alpelisib, a PI3 kinase inhibitor, and they're very sensitive to talazoparib, a PARP inhibitor that can target BRCA1 mutant cancers. So you can, in theory, now do this from top to bottom using all of these techniques. You can try and understand the disease that patient had suffered from, and better and now precision medicine approach to treatment. And so I'll end by showing you what happens now with the single cell sequencing. This is done all with bulk sequencing. Bulk sequencing and then organoid growth can identify mutations and therapeutic targets. But when you do the single cell sequencing, now you can really understand at real depth what's happening within this tumor. So we single cell sequence, we can identify fibroblasts, osteoclasts, lymphocytes, endothelial cells, and these different epithelial populations. And you can now break this down. And as I said, on the bone metastasis on one side versus the other, they have different levels of cells in there. They have different epithelial populations, different lymphocyte populations. They have different copy number aberrations between the different epithelial populations. These copy number changes aren't found in any of the normal cells. And then finally, we can now drill down into these populations and look at these different cell types. So this is looking at the fibroblasts. There are different populations of fibroblasts in this left and bone metastasis. This shows you the left and the right and what's going on with these different populations. And you find, for instance, hypoxic ones that are present and different between the left and the right. The epithelial cells show different changes in EMT programs and stem cell features, as shown here, and different between the populations of cells within the left and the right bone metastasis. And as I said, the immune landscape is different as well. So you have different populations of immune cells in that left bone and the left right. It shows an incredible heterogeneity. And obviously, the more we do, the more we're going to find. And the key point is, how can we reduce this complexity and try and understand the key features that are important for treatment of that bone metastasis? So to summarize, in the breast cancer cell lines, they show genetic heterogeneity with multiple subclones. Lots of E-cadherin alters the transcriptomes with cells clustering independently. Then ILC cell lines, tumors, and IDC cells with loss of CDH1 show increased IRF1 levels and activity. These patient-derived organoids, we've generated over 20 ILC organoids, which generally lack E-cadherin and have CDH1 mutations. These patient-derived organoids maintain the heterogeneity of the tumors much more faithfully than the cell lines do. And this is clearly higher than you see in cell lines. And some of these patient-derived organoids clearly express ER and have E2 inducible gene expression. For the mixed IDC ILC, as I said, we've done 3 of these now. You have to do this fresh, and we don't see many of these, so this is not trivial. But the data is also not trivial. We're seeing an incredible complexity of cell types and heterogeneity, and it's very hard to understand, to ascribe features to individual cells. But we do see low CDH1 cells, which we think may be the lobular component compared to the ductal component. We have a lot more to do to try and understand this heterogeneity. And finally, the ILC bone metastasis, we see evolution and gain of both genomic and transcriptomic changes, such as the BRCA1 mutation. And some of these may lead to unique therapeutic vulnerabilities. A PARP inhibitor may have been useful in that setting. This epithelial transcriptomic heterogeneity comes with things such as an EMT subclone that may be an aggressive subclone. And we see fibroblast and immune cell heterogeneity, even between an individual patient and metastasis on different bone sites in a single body. So we are super fortunate. This obviously takes a village to do this, and we are super fortunate to have a very strong lab and very strong collaborators. I've highlighted in red the people who had key roles in the presentation I talked about today, but in essence, everyone is committing and helping to all of the projects that we do in the lab. We work very closely with our physicians, and we have our autopsy team as well. We work with lots of other outside collaborators, and all of this is made possible by funding through government foundation or foundations. We work, as you know, very closely with the patient advocacy community. And finally, but not least, we need to thank the many patients who donated their tumor tissue. Obviously, we couldn't do this research without that, and we hope that their donation is going to improve the lives for people in the future. Thank you.
Thank you very much, Adrian. This was a beautiful presentation. It's also beautiful to see everything you have already achieved in terms of, taking it a step further to generate actually resources for experimentally investigating the disease. It's really beautiful to see this ILC cell line encyclopedia, also to see the PDOs that have been generating, and also the rest of your data, which are extremely convincing. Before I go to the questions of the audience, I also had some questions. So I think the great interest of the last results you showed is because you were able also to compare data within the same patient. And I think that this is also actually the way to go, because we know that sometimes there's a heterogeneity in terms of response of disease evolution. And I would like actually you to comment a bit more on that.
Yes, you pointed this out actually in your keynote lecture when you showed the slide, and you said, be careful, these are not matched. And we have spent a lot of time and energy because we really believe, I think the best analysis is a paired analysis within a patient, because then the unique changes are likely related to the progression in that patient. And the disease is so heterogeneous, particularly metastatic disease. It's very hard to understand, the forest from the trees in that setting. But as you know, this is incredibly difficult, because if it recurs 20 years later, who has that primary from 20 years ago? We were very lucky in Pittsburgh to have a population that doesn't move around very much, and that's what gave us this opportunity. But I think you had said this, and this was said in the advocacy meeting this morning. It's very hard to do that alone. I mean, I think you need consortia like you guys have been talking about, a consortia to pull together samples, to pull together clinical records. I think that's really one of the only ways you're going to get the N large enough to address this. We've had a lot of our own small studies, but in the end, it takes more than that.
Yes, and also another comment I think is that having all these cell line encyclopedia is also great, but I think that what we need to be careful is also to have the right annotation that goes actually with these cells, because having just the cells with their molecular alteration also might not be enough. And I think that that's a general effort, and that's something that you might be working on prospectively. But I think.
So the thing I would say about that is I totally agree. I feel those cell lines, most of them were made 30 years ago, and people weren't thinking about that 30 years ago. And so we can characterize them at the molecular level as much as we like. I mean, people use cell lines because they're an easy to use resource. They're relatively cheap, but they have their limitations, and people accept that. The organoids will be very different because, as you know, we're doing that prospectively. We have full clinical records. We understand everything about those patients. And I think they will be a better resource for research. Now, they're very expensive to use. That's the real problem. And they're not easy to grow either. And they grow very slowly. So they have their own series of challenges. But I think they're a better, more faithful representation of the disease than the cell lines by definition.
Yes, I fully agree. And sometimes actually just with one patient, we can learn about resistance mechanisms, which can be also extremely promising. Totally. So just some questions from the audience. So one question is actually, how does one go about donating preserved pleomorphic lobular tissue or arrange for possible future tissue donation actually?
Yeah, so we are very focused on pleomorphic as well. We've sequenced the numbers of pleomorphics because that's one of the most aggressive diseases and has this very high rate of HER2 mutation, which is also very interesting, which you commented on. it would be great to have a centralized way to send tissues to, but again, this is, as you know, a challenge. You know, there's challenges over legal issues and university issues. We've discussed with many groups having a centralized tissue collection. That's not really possible. I think hopefully people can seek out their local group and then hopefully donate to them. If tissue is cryopreserved, live cryopreserved, we can make them in the future. We know we can do that. So I think there'll be even better things coming down the road. As long as we keep the tissue, we can do things in the future. Yes,
and I think we might go, better in the coming months and years. So then also there was a comment with regard whether, again, you mentioned that PDOs are extremely expensive, but to have really a realistic approximation about the cost, both money, personal time, how long does it take to develop, to maintain these PDO models, and how feasible is it to go, for example, for smaller labs and things like that?
So we have a very high success rate, 80% of them. The issue is getting a good collaborator as a surgeon to get to the tissue and de-identification and those things. But once you have them, the reagents are very expensive. You know, generation of a PDO, I don't want to give a number. I know it's one of the people who's from the lab could give the number, but I probably give the wrong number. But it's, you know, if you want to buy it from ATCC, as an example, they charge for a cell line $500. For a PDO, they charge $3,500 to buy a PDO. So that gives you a heads up of the cost of generating them. But small labs can generate them. It's time and effort, basically.
Yes. Then I think there was another question also for people thanking you for the very clear talk. And a person was wondering if there were some differences at the microbiome level, actually, or that you have been noted.
Yes, so someone in our group has looked at that. Funnily, that's an interesting question, because someone in our group you can do that from the single cell data, because you have reads coming, you map the non-human reads, and you can identify the microbiome reads. And so they've been taking our data and identifying the unique microbiome in them. One wonders, is that a true microbiome from the patient, or has that come through the lab or sequencing? And there's been lots of controversy over that. But I do think there's, you know, the microbiome is, you know, it's a major area. And if her data is true, our collaborator, then I think that will become quite interesting.
Yes, of course. And of course, it will require some dedicated studies with the right protocols for that. Okay, then there was a question, I believe, from a patient that is asking whether a metastatic breast cancer patient could help by asking for her original tumor to be transferred from the first institution to current, or to be kept. And also the question to know whether tumors are routinely discarded after a certain amount of time.
Yeah, so that's a great question. And I think that's where patient advocates can help in enforcing this issue. I think many people are unaware that tumors are discarded after a certain amount of time, after I think it's 7 years in the US. I don't know how that is in the UK or in Europe, I should say.
Longer, much longer.
But I think patients own their sample and patients own their data. There's a big push in the US at the moment to get the data, electronic health record, to the patient. It's the patient's data, yeah, it's the patient's tissue. And so patients can push for that. And we've had patients, you know, in our autopsy program, a lot of it is the patients pushing to be on the autopsy program. And if they're in the wrong hospital system or the wrong insurance network, you know, we can sometimes figure these things out. So I do think this is a great role for advocates in advocating for tissue research, because it's only through research on the biosample do we ever make any progress. Yeah, that's the way we've made everything we have has come through that. Yeah, so I think we now have so many better tools and techniques to use the tissues than we did have 20 years ago, that they're so much more valuable now, that if people can push for them, yeah, they should try.
I fully agree. And I think that this is the way forward, to have much more of this patient partner research, actually, to have them much more involved, and also to help decide about what is going to happen, actually. Yeah. So one last question before we move to the last speaker. So you showed also extremely interesting data on these tumors that we do not enough talk about and still do not know what to do, like the mixed ductal lobular tumors. So I was wondering whether also there you were doing some more spatial analysis to really look at the distribution of these different markers.
Yes, so we're just So we have lots going on in the mixed. That's quite an interesting area that we got funding from the Dyname Foundation to do. And so Steffi has led a study of the unique clinical characteristics of the mixed, because it's really a spectrum of anything that doesn't fit in those two buckets goes into this middle one, yeah? And there's clearly differences in there. And so we do the clinical characteristics, and then we are just about to start the spatial sequencing, literally in a few months. And I think, or the spatial analysis, I should say, but it's spatial sequencing. I think that's really, I hope that will be easier to understand than the single cell sequencing, which has been complicated.
I believe it should. Okay, and then one last question that I forgot. So a question to know whether metastases still look lobular under the microscope.
You know, I saw someone ask someone earlier that today, and I thought, you know, yes, sometimes they still look lobular and sometimes they don't. It's, as you know, I think someone said this earlier, I think it was one of those first pathology talks. This seems to be back and forth between some of these phenotypes. And I think that's incredibly complex. You know, we have some that are going from one to the other and some that are moving backwards. I think we need to do a lot more on that. We need to look at the ecadherence status in the metastasis and then the histomorphology of the metastasis. I think that's a great project that we could do together.
Okay, fantastic. So thanks a lot for your presentation and for this Q&A. Thank you.
Thank you.